In October, I attended the 3-day virtual Firefly Symposium that was hosted by the Xerces Society, and one of the presenters was Dr. Avalon Owens, a research fellow at the Rowland Institute at Harvard. I found her presentation highly fascinating, and I invited her to speak to the Maryland Entomological Society. She said she would be delighted, which leads us to tonight. Dr. Owens holds a BA in integrative biology from Harvard University, an MS in entomology from National Taiwan University, and a Ph.D. in biology from Tufts University. Dr. Owens' laboratory studies the laboratory studies how organisms and ecosystems cope with anthropogenic light pollution. The lab studies the impact of light pollution on organisms and ecosystems with a focus on firefly conservation and moth evolution. The lab is particularly interested in the ecological costs and evolutionary history of flight to light behavior, positive phototaxis, most famously exhibited by moths and other insects. The lab uses a range of methods spanning the fields of spectroscopy, stereophotogrammetry, remote sensing, computer vision, entomological collections, pest management, animal behavior, and conservation genomics to improve the understanding of both positively phototactic insects and the increasingly bright light environments in which they live. So the bottom line is the Owens Laboratory studies how organisms and ecosystems cope with human development.
Wow.
And I will introduce Dr. Owens, and you can share your screen.
All right. Does that show up okay for everybody?
Yeah. That is good. Let me make one more adjustment here. Okay, we are good. And everyone, please mute your microphones so we don't hear you out in the middle of Avalon's talk tonight.
All right. Well, thank you so much for having me, everybody. I'm really thrilled to be here. Seems like a really nice audience, and I bet we'll have a great Q&A session at the end, especially once I get to the synchronous firefly part, which comes in the second half. So first, we're going to learn something about moths, which I think will also be of interest to you for reasons that will become clear right now. So I'll just start. Right now, the world is changing at an unprecedented rate, and organisms face a range of really rapidly intensifying anthropogenic threats, including deforestation and climate change and invasive species. This is kudzu, urbanization, as well as other things. And as a global change biologist, I want to understand how organisms cope with changes of this kind. Why are some able to thrive in the Anthropocene while others face extinction? And to understand this, my research group focuses on organismal responses to recent change in perhaps the most foundational resource on planet Earth, which is light. Light is the source of all life on Earth. It's the foundation of our food webs. It's the major way in which almost every species experiences its environment. And it's a reliable signal of passing time that has been constant for all of evolutionary history until now. We are rapidly and radically altering natural patterns of light and dark. And by studying how organisms respond to this unprecedented selective pressure, I hope to gain novel insight into the future of life on Earth. I should note that this threat is not limited to large urban centers like San Francisco or Baltimore. As of 2014, 23% of the habitable land surface of the Earth was unnaturally bright at night. And our nights are only getting brighter over time, about twice as bright in just the past decade. Over half of all insects, that would be most insects, are primarily active at night, and our lights are completely transforming their world. They struggle to cope with this change in particular because it has no natural analog. And I spent a lot of time studying whether fireflies will survive given how difficult we're making it for them to communicate. But more recently, I've turned my attention towards moths because moths will spend entire evenings, a significant chunk of their very short lives, throwing themselves at artificial lights over and over again, circling them or sitting stunned beneath them. This behavior is often fatal. You can see on the left here insects combusting in the heat of a floodlight. And just for a second, can we get that person to mute their microphone?
Yeah, Fred, can you mute everyone, please?
Ah, beautiful. Thank you. Okay, where was I? So on the left here, we see insects combusting in the heat of a floodlight. So not a great thing for them to do. And insects don't always die because of this, but even if they don't, they have lost entire evenings of time that they could have been, foraging, mating, ovipositing, whatever, what have you. And while the exact proximate cause of this behavior is sort of remains to be determined, what we know is that it's clearly some sort of evolutionary accident. These organisms are unable to cope with a novel change to their environment. And it's natural then to wonder, okay, what are the long-term consequences for biodiversity? So I want to understand how nocturnal insects are doing in response to artificial light. However, this turns out to be tremendously difficult to study because we use artificial light to understand how nocturnal insects are doing. Entomologists use light sheets and light traps to systematically survey insect communities, and we've done this for a very long time. When we don't see moths at lights, we get concerned. In recent years, scenes like this have sparked fears of what we call the insect apocalypse, which is heightened by anecdotes about not seeing moths swirling around streetlights or plastered on lit windows or caught in car headlights. The logic goes, we used to see many moths at streetlights, and now we don't. We typically assume that a light trap will catch a constant proportion of the surrounding population. And in this example, that proportion is 50%, and the moths not caught by the light I'm showing in gray. Then data like this imply that we've gone from 6 moths to 2, which we might call the apocalypse scenario. An alternative explanation, however, is that today light-attracted moths are spread among more lights, and each individual light is less attractive than it used to be, set up against all of our light pollution. In this scenario, the moths are still there at lights. It's just that the lights we use to monitor them are being outcompeted. Another alternative explanation is that moths today are simply less attracted to lights than they used to be, because flight-to-light behavior is highly maladaptive, and they've already had several hundred generations in which to figure this out. In this scenario, the moths are still there, they're just not at the lights. To evaluate which of these scenarios is responsible for the patterns that we see, the first thing we want to do is to compare what the light traps are telling us to what other traps are telling us, to see if other kinds of traps are showing similar declines. The problem is that people rarely use other methods of systematically catching and counting moths, except in agricultural settings, where pests like the corn earworm moth have been tracked extensively for decades. And so I collaborated with some folks in the extension space on the following study. The state of Delaware has a monitoring network with redundant traps, pairs of redundant traps. So this is one of 11 farms in that network, and it has a blacklight trap, which is placed right there, and a pheromone trap, which is placed right there, less than a kilometer away. And both of these traps are checked every couple of days, and the number of moths within are counted and recorded. And this has been going on since 1998 with the same blacklight, with the bulb replaced every year, the same brand of pheromone lure, and the same general location. And this, the analysis part of this study was done by my student Ian Battles, and it's published in the Journal of Insect Conservation if anybody wants to give it a look. So here I'm going to show how the number of moths caught in these traps vary over time. The pheromone trap data look like this. We see no major changes over the past 25 years. Now, if the blacklight traps still work as well today as they used to, we would expect this line and the line that I'm about to show to be parallel. The average might be higher or lower because of differences in how these traps work. Of course, one involves sex pheromone, it only attracts males, the other light, and so forth. But the percent of the population that's caught by each trap should be constant over time. That's the fundamental assumption that we make when we do long-term monitoring. Instead, the blacklight trap data look like this. They start at around the same place, but then diverge rapidly and are not at all comparable today. Just down the road in New Jersey, a similar monitoring network has been running since 2013 with redundant traps. And it turns out that 10 years is enough time for these lines to diverge significantly, again, in the expected direction. And in fact, if you look at only the most recent 10 years of the Delaware monitoring network right next door, the situation looks almost identical, even though these are completely different monitoring networks run by different people in different states. It's not necessarily surprising to me that 10 years is enough time to see a significant decline in blacklight trap effectiveness, because I said this earlier, in just the past decade, the night sky has become twice as bright. If you could see 250 stars in the sky at night as a child, as a college student, you'd be able to see less than 100 on average. And that is thanks in large part to the growing popularity of super cheap, super bright LED lighting. A generalized additive model shows that blacklight and pheromone traps only really started to diverge around 2010, which is around the time that Delaware began replacing older streetlights with modern LEDs, which could have increased light competition. And we've run a couple of models at this point looking for those environmental effects on these counts using satellite imagery. And they have been inconclusive for reasons that I could describe later if you're interested. So a bit of a question mark there. But this pattern could also be caused by rapid evolution, because you could imagine if 25 years ago we had a population of moths where most of them were attracted to light, but a few of them weren't, a pheromone trap catching a third of this population would get 6, whereas a blacklight trap would be catching only the white moths, a third of those would be 5. There's not a huge difference. But as the proportion of the population that isn't attracted to light increases because the costs of being attracted are so high, then the pheromone trap stays about the same, while the blacklight trap counts go down. And then today we might see a situation like this, where the pheromone traps are catching 10 times as many moths as the blacklight traps. If this is true, if evolution is to blame, then we can sequence moths from traps today, pheromone traps and blacklight traps, and compare the genetic composition of the moths within to see if there are differences between them. So we've done this, and the short version of the long story is we don't see a single nucleotide change, that some very strong marker of divergence, but there is a little bit of a difference in regulatory elements, and we identified a pathway. It's called PKC. It has to do with light adaptation that is under positive selection only in the pheromone group. So it's pretty exciting, although more to be learned. And before I go too much deeper into that, I just want to wrap up this section to summarize, we saw that the blacklight traps in Delaware and New Jersey used to catch a lot of moths, and now they don't. Is it because the number of corn earworms has changed? No, because the pheromone trap counts remain high. So it could be because the blacklight traps are less attractive than they used to be. Moths are choosing to fly towards LED streetlights or other light sources instead. Or it could be because the moths that fly to light sources frequently die before they can pass on their genes to the next generation. And perhaps selection is eliminating the flight to light response. A big difference between these two scenarios is what happens when you turn the lights off. If the moths are still attracted to lights, they'll come back. But if some of them aren't, then when we rear different populations in a common garden, we might expect to see a pattern like this, with rural moths today less likely to fly to lights than their historical conspecifics, and urban moths even less so, because they've had more generations in which to adapt. No one has ever compared historical and modern behavior in this way. And the reason is that historical reports of moths at lights are pretty common, but actual numbers are very hard to come by. However, In 1969, which for my purposes we're calling the distant past, Paul Stewart and colleagues measured the percent of corn earworms that flew towards a black light at different distances. And so we decided to replicate this experiment as perfectly as possible 55 years later to look for behavioral change over time. Stewart et al. Found that 87.5% of corn earworms, same species, moved towards a black light at 6 meters distance. That's it. He also found a lot of other things, but that single data point is the only thing I'm interested in. My question is, will corn earworms from urban and rural backgrounds today still fly towards a black light at 6 meters distance 87.5% of the time? And to do this, a lot of this work was carried out by my postdoc, Wei-Ping Chan, as well as my student, Imogen Dashak. And we also worked with a big team of collaborators who helped us collect caterpillars from sentinel corn plots that span the entire corn earworm overwintering range. And the sites we collected from included some very light polluted areas like Corpus Christi, Texas, and Raleigh, North Carolina, as well as some very rural locations like Macron Ridge, Louisiana, and somewhere else in North Carolina. And we reared these caterpillars to adults in the laboratory, and then we took them out in the field. In 2023, we took our populations to the Harvard Forest in Petersham, Massachusetts, and released them into these tunnels, which we built to the original specifications. And we were looking to see, according to the original methods, when we released the moths in the middle compartment, how many of them moved towards the black light in the center here versus away from it. We did this at night, obviously, so that's what the black light looks like through the tunnel. There's mesh on either end of the tunnel, by the way, that keeps the moths inside. And then in 2024, we built 2 more tunnels, reared way more moths, and took them to a remote cabin in Lakeville, Maine, which is one of the only places in the entire eastern United States with pristine night skies. And that's what it takes to have an open area as dark as it was in 1969. This is a video of our experimental protocol. So as the sun sets, we would go out, set up the boxes, and then release the moths, give them a little while to settle, turn on the light, give them time to move towards it or away from it, and then close inner doors in the tunnel and count which moths were in which compartment. And this is what we found. So here I'm showing the collection sites on the x-axis, and they're plotted according to the light pollution levels within a 5-kilometer buffer around the collection site. And then the y-axis shows the percent attraction. 50% in white there means no choice. They had no preference one way or the other, going towards the light, away from it. But we did find a significant preference. It was about a 66% attraction to the light. And that was the same across urban and rural genetic backgrounds. There was no difference there. However, this current average was different from the historical value of 87.5%. So this was pretty exciting. But to make sure that we were seeing a real difference, we ran the experiment again with new moths at this pristine dark sky site. And there we recovered the historical value of 87.5%, almost exactly. It was eerily close. So, wow, that suggests that this is not about the urban, rural, or historical background. It's not about the genes. These moths are all behaving pretty identically, but it's about the environment. So something about the environment. What could it be? Well, Petersham, Massachusetts, where we went the first year, and Lakeville, Maine, the second year, they're pretty different in terms of upwelling radiance, which is what the light that the satellites see, as well as sky glow, that reflected light, the glow of cities, which is modeled from those data. And overall, as you might expect, it was very, very dark in Maine, not so dark in Massachusetts. It was darker in Maine where the moths were more light attracted. So it might seem like a closed case, but we shouldn't necessarily expect to be able to see what a moth can see from a satellite. It's rarely that simple, unfortunately. So like I said, the site in Maine was open to the sky. We had a pristine night sky, and you can see that in these long exposure images, which are taken from inside the tunnel looking out. This is the view of the moth from inside the tunnel, looking away from the black light. So you can see that open sky. And then our site in Massachusetts was in the middle of a forest, and you can see that here. These images were taken with a calibrated camera, and by averaging them, we can quantify the absolute radiance at varying elevations within the field of view, within this field of view. So elevation there refers to this image. And we found that the site in Maine was uniformly bright from top to bottom. So natural light from the night sky reflected off of the grass and made a nice bright scene. Whereas in Massachusetts, it was darker, but also much less uniform. So in the forest, the trees were blocking out the night sky, but letting in light pollution at the horizon. And what that means is it was darker overall, where the moths were less light attracted, which suggests that that response is not about the absolute contrast of the black light against the background, but instead has to do with the complexity of the visual scene. And this is something that we're exploring further in the laboratory right now. Ask me about that later. But I'll just wrap up this section by saying, if we look at all of the data from the Delaware Monitoring Network for the last time, we can see that the corn earworm seems to be doing pretty well overall. It's not of conservation concern, in my opinion. But if we only had data from blacklight traps, we might have a very different impression. Note that this graph is on a log scale. Blacklight traps catch so few corn earworms today that some of my collaborators don't even bother using them. But insect conservationists use them because they're all that we have. So for most moths of conservation concern, we only have data from blacklight traps. Yet these data are conflated, which makes things really complicated. And I don't have answers except to say we should figure this out now, because automated methods of monitoring moths and other nocturnal insects using lights and cameras and computer vision processing are becoming incredibly popular. Community science efforts as well. You can see here on this map, each of these locations is a place with a long-term monitoring project based on taking images of moths at lights. So we should make sure that these data are useful and usable into the future. I could go on, but just want to say, it seems like threats to moths are intensifying. Light pollution is one of these. And at the same time, our method of understanding them is breaking down because of light pollution, which leaves me with a lot of questions. But I don't just want to understand how insects respond to light pollution. I also want to make things easier for them by promoting practical solutions that protect nocturnal insects and their habitats. But for sustainable lighting policy and advocacies to succeed, we need buy-in from the general public. And I know everybody in the Zoom call loves moths, I'm assuming, all of us. But, you know, gotta keep it real. We know that everyone loves fireflies. Fireflies can act as flagship species to inspire dark sky conservation efforts, as long as the sustainable lighting practices that work for them work for other species as well. So if you've never seen a firefly, I can't imagine, but if you haven't, this is what they look like. Each string of flashes that you see in this long exposure photograph is an advertisement that's produced by a patrolling male. And you see one on the top of the screen there, a single male flying a path, emitting a light. He's looking for a response from a female on the ground, hopefully a response flash from a receptive female conspecific, and not an imitation from a predatory female in a separate genus who wants to lure him in and eat him. Firefly, what do I want to say about this? Everything in a firefly's life is very dramatic and very visually, it's very important for them to see each other. Their ability to see is like crucial for everything they do. So let's talk a little bit about how they do that. I'm going to show you, this is a spectrum. So on the x-axis are wavelengths of light, and the y-axis is, in this case, an amount of absorption. So these are the colors of light that the firefly eye absorbs. Fireflies have only two types of photoreceptor. One picks up short wavelength light in the UV range, and the other one picks up long wavelength green light. Specifically, the color of conspecific bioluminescence, which looks like this. It's an almost one-to-one match. The light that they put out is the same color as the light that they take in with their eyes. What this means is fireflies are effectively red-green colorblind. So a scene that looks like to us like that on the left would to a firefly look like that on the right. Fully nocturnal fireflies tend to produce green flashes, and green shows up as well as any other color against a fully dark background. And I've represented a background here. This is the spectrum of a moonless night. But because fireflies active at dusk occupy a bluish light environment, which was made greener by the reflection of light off of trees and grass, these species have shifted their flash color and their visual sensitivity to yellow. They physically filter out environmental light to maximize the visibility of conspecifics. And to put that in another way, firefly flashes are really easy to see in the dark. They're harder to see against most colors of background light. And to solve this problem, fireflies have not evolved some incredible capacity for color discrimination, but instead filtered their vision so that they only see the one color that matters. And this adaptation works great in natural light environments. It has for over 100 million years. But what about artificial ones? Like I said, actually, just keep moving. So the first thing that we want to do in exploring this is to test experimentally whether firefly flashes are actually less visible under artificial light. And this is something we did working with the species Photinus greeni, which has recently been renamed. So I should update this slide. It's Photinus jamesi now. That was a major blow for me. So I'm going to show you a video of their courtship dialogue on the right here. So you'll see a male flash inside that circle and a female will respond inside the other circle. So in this species, the males do a double flash, 1, 2, and the females respond after a time delay, and then it goes again. So the male will go 1, 2, and the female responds. So we use this video to get a waveform of a female response flash, and then programmed 2 LEDs to produce that flash after a time delay. So that when we saw male fireflies flying around advertising in the field, we could respond by triggering both LEDs with a shared button. And the male in question usually got pretty excited and would come over to investigate. When one of our females was illuminated by an overhead light, males approached the unilluminated female 96% of the time, which suggests that artificial light does make firefly flashes much less visible, or at the least less attractive, which in fireflies is probably the same thing. So what is a firefly supposed to do about this? Well, this model of firefly communication in natural light environments can inform some predictions about how they might adapt to unnatural light environments. Here's what the spectrum of sky glow, that haze of light pollution, looks like, by the way. It tends to be kind of red. And my first thought on putting this model together was, well, maybe dusk active fireflies and habitats with a lot of sky glow could shift the color of their bioluminescence back to green in order to increase its contrast against a red background. Data from the common eastern firefly, Photinus pyralis, show that flash color does vary quite a bit within this species. The color of the points on this map are exaggerated. It's about 10 nanometers, which is not a lot to us, but it is a lot to a firefly. However, when we plot that against the intensity of local sky glow, we don't see a strong relationship, which suggests that there hasn't been enough selective pressure to cause the kind of evolution that I was expecting in any case. Okay, well, there's another thing that fireflies might do instead, and they could even do this on an individual level via plasticity. They can just make their flashes brighter, which would make them more visible against any color of background. We know that urban birds will sing more loudly to be heard over noise pollution. Maybe urban fireflies flash more brightly to be seen over light pollution. So to test that, we recorded the courtship dialogues of Photinus obscurellus fireflies in the laboratory. In this contraption here, where we have these 2 chambers, we separated a male in the top from a female in the bottom using a transparent barrier. And the 2 would flash back and forth for up to an hour while we cycled through 5 colors and 2 intensities of downwelling light. That's what you see at the top there. Then we recorded the whole exchange using a low light camera, which had a green bandpass filter attuned to their flash color, which blocked out the light that we were adding to the enclosure, especially from our red and blue treatments. We then used computer vision to estimate the intensity and duration of each flash that we caught on film, where the intensity is the number of pixels of the flash and the duration is how many frames it appears in. And we found that there's a lot of variation, but on average, flashes captured under red or especially blue light tend to be a little bit longer and a lot brighter than those captured in darkness. So these here are the means of each of these values. All right, that's a great adaptation, but is it enough? The final possibility that we have to entertain is that a light-based communication system just can't survive on a light-polluted planet. And let me show you a video from this experiment, and maybe you'll start thinking about it in a similar way. So in darkness, the male on the top flashes a lot, and the female in the middle of that circle responds every time. So he's flashing and flying around, she responds, he flashes, she responds, and then we turn on a cool white light, and you see the male in the middle of the compartment. He's still flashing. The female's in the same place in the middle of that circle, but she doesn't respond. And he keeps trying, but no luck. And what this looks like graphically is the males flash about 10 times a minute in darkness. Females respond almost every time. Under these colors and intensities of light, the males flash about half as much, and the females sometimes never respond at all. Under bright amber light, we never saw a single flash from a female. She might not be able to see the male. She might find him less attractive. The outcome is the same, which we call courtship failure. However, we tested all these colors and intensities of light, and we did find differences in the female responsiveness. It looked like dim blue and dim red were less disruptive than other colors, which suggests a possible way forward, what we might call a sustainable lighting solution. So the last thing that I want to do today, depending on how much time there is to take that idea out of the laboratory and into the field. And finally, what we've all been waiting for. Fireflies in the species Photinus carolinus will flash in synchrony all together about 5 to 8 times, and then they all go dark more or less. There's a long period of a wait, and then they start up again. This species is found hold on. This is found in the southern US. So this is a different kind of synchrony than the fireflies in New Guinea. And we can talk about this after. It's a loose synchrony. It's very beautiful. And these displays will attract people from all around the world to remote parts of the United States, including Kellettville, Pennsylvania, which is home of the Pennsylvania Firefly Festival. When you put humans and fireflies in the same place, something interesting happens. Visitors have to see where they're going for safety reasons, right? These events happen in the woods. They're often on unpaved trails. And so visitors need access to some sort of light. But the fireflies don't want that. So the Pennsylvania Firefly Festival is trying to find a middle ground that keeps people safe and fireflies happy. And for decades now, firefly researchers have advocated the use of what we call firefly-friendly light sources when out in the field. And that means lights of a color that fireflies don't see very well or don't react to. But we can't agree on what that color is, red or blue. And amber or orange, yellow, I call it amber, this has emerged as a third option. It's often touted as a sort of insect-friendly light, generally insect-friendly. So in 2019, we set out to explore the relative costs and benefits of each in the following way. So these are the grounds of the Pennsylvania Firefly Festival, and we worked alongside this forest edge where fireflies from the woods spilled out into the yard. We set up 4 plots with LEDs, floodlights of different colors. We have blue, amber, red, and a dark control. They're all equally bright, and then we would randomize them each day. So almost every night of the firefly season, from 10 p.m. to midnight, I and my student Caroline Dressler, as well as Morgan sometimes on the right there, would survey these plots in random order, counting the male flash patterns produced within each plot during a 1 minute observation period. And we also used sticky traps to sample the other insects that were swarming around our lights to get an idea of how the lights, how are the lights good for fireflies? How are they good for other insects? And this is what we found. So on the x-axis, I'm showing you a transect that extends from the forest edge. And on the y-axis are the number of flashes that we saw in that section of the transect. So in darkness, we saw a pattern like this. There were a lot of flashes right next to the forest, and then sort of as you went out into the field, they fell off just because the species prefers to live in the woods. When we put an amber light right in the middle there, we saw this enormous drop in flash activity, and we saw similar but less dramatic drops under blue and red light. All three colors significantly impacted the firefly display in the middle there. And none of them had an impact near the forest, and only amber and I think blue, yes, had an impact far over here in the field. So that sort of recapitulates the results we have from the lab, where all lights are disruptive, but amber is the most disruptive. And now if we talk again about flight to light behavior, coming full circle, these are data from the sticky traps that we put under each light. The sticky traps in our unlit lights caught almost 50 insects that were very unlucky. They were just passing by. And our blue, amber, and even red light caught significantly more insects. Of the three, amber and blue caught the most insects. And all of these, yes, amber and blue were most attractive. And in case you're curious, these were mostly mosquitoes and gnats. And so we could talk more about this, but that might explain their relative attraction to yellow versus some other insects. We also caught 3 fireflies, 1 firefly on an amber sticky trap, 1 on red, and 2 on blue. But the thing to take away is that in both cases, red is what we might call the best of the bad options, and amber is clearly the worst. So wouldn't it be unfortunate if amber lights were only becoming increasingly popular as dark sky friendly, sustainable lighting solutions? We've spent a lot of time in my field trying to find some sort of magical color of light that only humans can see, that has no serious astronomical or ecological impacts. But I think by now, most of us have realized that we might be attracted to lights like this, but they're distracting us from better alternatives, highly metaphorically. So let me check the time. I'm making good time. Do we take the temperature of the room? Should we have a Q&A, or is there interest in talking more about sort of dark sky mitigation methods and which ones are best for insects or otherwise?
If you want to keep going for a little bit, Avalon, I think it'd be okay.
Okay, this part can be fast. I think
Okay.
it's nice take home because maybe you come away from this, thinking more about artificial light as a threat to insects, and there's changes you might want to make in your backyard. If you get interested in dark skies, or we call it dark sky advocacy, or light pollution advocacy, dark sky conservation, light pollution, etc. Pretty quickly, you're going to hear of Dark Sky International, which is it's an international nonprofit which aims to promote dark skies however possible, and they have what they call the 5 principles of responsible outdoor lighting, which are sort of in order. It goes like this: a standard bright blue-white unshielded street light, not great. You should shield it so it points down. Make it warmer in color. So they do promote amber lights a lot. Oops, sorry. Keep it really dim, and then put it on a timer or a motion activator. And these recommendations, Dark Sky International was founded by astronomers. A lot of this stuff is coming from astronomers, and astronomers are really interested in preserving the night sky. They're less focused on what's happening on the ground. In fact, A member of my very chapter once said, it's dark sky international, not dark ground international. So while our interests kind of overlap, there is some difference there. And these recommendations are great for preserving night skies, which is great in general, but some of them aren't that impactful for insects. So we just talked about color, right? So they say you should use a warm color bulb whenever possible. And it's true that insects are more attracted to short wavelengths of light generally. So that's why we saw more insects at the blue and the amber lights versus the red one. And it's also why we use UV light to attract moths, right? It's a very short wavelength. But normally when people are talking about streetlights, they're talking about what we call Kelvin, like color temperature. So like a blue white light or a warm white light. And there's been a lot of studies on this. Anything within this range does not make a huge difference for the number of insects that your light attracts. It could have an impact on fireflies, but even in the laboratory study, the warm white and the cool white light, they responded to them in about the same way. So there's not really much, I think, point in focusing on color as an entomologist. Then we have the use of, oh yeah, and like as proof that a warm light can be pretty detrimental. These are, I didn't say this earlier, but these are mayflies that are attracted to the lights over this bridge. And so they're ovipositing on the bridge because of the way the light reflects off the surface instead of in the water. And it's sort of horrible. And these are pretty warm amber lights. And you can also think of a moth flying into a flame. It's a pretty warm, literally, and also in color, a pretty warm light. So then we have the use of timers. So a timer turns off a light at a certain time, usually like 11 p.m. or midnight, after people are likely to have gone to bed. But most nocturnal insects are active before midnight. Like I think the exception are some moths, but I think a lot of the insects that we see the most at lights are attracted right after sunset when the light would still be on. So I'm not fully convinced that timers make a huge difference for insects. And this is just showing that in moths, like we could talk about activity timing, it's pretty interesting, but the time at which they mate is different than the time at which they forage and so forth. But a lot of the good stuff is happening before a timer would turn a light off. So then we have dimming a light to make it less impactful. Surely there's no way this can't help, right? I don't think it's necessarily bad, but I think we really overestimate how much it can do, because when we dim a light, it's very dim to us, but moths and other nocturnal insects, fireflies also, their eyes are thousands of times more sensitive than ours. So a light that's dim for us is pretty bright to them. And if we were to make it dim enough so as not to disturb insects, it would be so dim that we couldn't use it. So there's not much point. And then the last thing that they suggest is this idea of shielding. So constraining a light so it only points right at the walking path. And I was pretty suspicious of this at the beginning because, pointing the light down, if the insects are on the ground, I didn't think it would make that much of a difference. But there's a lot of emerging work showing that it does. This is a fantastic study that came out in Germany where they redid these lights in a public park. So on the left is the lights before they fixed up the fixtures, and this is how it looks to us. And then this is a false color image that shows where the light is hitting. And then on the right here, they put on these custom fixtures that were like customized to each light to make it only hit the sidewalk and not go anywhere beyond. And so you can see they've just like totally cut off all of the spill light. It's really focused on the walking path. And by doing this, they were able to cut the number of insects attracted to these lights in half, which is way more than any dimming or changing of color has ever done in any study that I've seen. So it seems to be a pretty useful thing to do. And that's great because it's also easy and it doesn't cost much, if anything, to just make sure your light is pointed right at the place where you need it to be pointed and not out onto the verge, the yard, etc., the neighbor's window. But the number one thing that they recommend, which I also recommend, is that you should turn off lights that you're not using. light pollution is defined as light that is wasted. So as long as a human is benefiting in some way, is able to navigate due to a light in their environment, I think that doesn't count as light pollution. But 99.999 most of the lights outdoors at night are not being used by anybody, um, seriously. And so that turning off things like decorative lighting, facade lighting on buildings, under bridge lighting, light coming out from windows without curtains, path lighting, that all the lights that are used for navigating can be put on motion activators so that they're on when the people are there and not when they're not. And then the other lights can just be turned off. And that is going to be, that's the control scenario. That's the best for insects. So just writing that out, that would be the first thing to do is to remove unnecessary lights, and then to install motion activators, and then use shielding to keep them confined to a small area. And then you can dim them, and you can put them on timers, and you can even change the color maybe, although, we could talk about color. Okay. And with that, I'm ready to take, let me thank some people first, actually. So sorry. Tons of people to thank. So my PhD advisory committee supervised the Firefly work. And I also worked with some collaborators and a lot of really great students on this, and got funding from some fine local nonprofits, as well as the Robert and Patricia Switzer Environmental Fellows program, and PEO International. And then the Xerces Society for Invertebrate Conservation, you all are probably pretty familiar. They've been just such great friends on this, and so interested in and passionate about firefly conservation. And Dark Sky International is also really interested in this. And then my more recent work, these are some of my lab members. And some of my amazing students. We've worked with a ton of collaborators on these folks worked with us on the first project I talked about with the trend lines, and these on the second one with the replicating of the historical experiment. And with that, I am ready to take any questions.
Can you go back one slide, please?
Yes. Anybody you know?
Is this our Galen Dively?
That's the one. Dr. Dively. He was provided a lot of advice.
University of Maryland.
Yeah.
Right.
What about David Wagner? Is this the Mothman, David Wagner, or the one
David,
the Caterpillar book?
yeah, Dave Wagner. So he was on my PhD committee, and he's also, definitely an ally in the insect conservation field, and we've talked a lot about light pollution.
Right.
Avalon, when I was in New Guinea with John Buck and Jim Case studying the synchronous fireflies there, we had image intensifiers, so we could see everything that was going on around us, but by starlight. There's no extra thing. So I'm wondering if that might be something you could add to your list, because it doesn't take any extra light. It just uses what's there, and makes an incredible increase in visible light through the um, through the eye pieces.
Oh, interesting.
So maybe if they come down in price, I think those are running around 500 to 1,000. They'll surely come down in price, and you can try that maybe, and just check people in with one. Make them leave their uh, their credit card with you or something
It's like
until you get it
for
back.
getting around. So it's like you use it sort of like like it would be like a infrared like night vision goggles.
Yes, exactly.
Yeah.
Yeah.
Yeah, I think yeah. Really what we need for people walking around at night is just to carry a flashlight and then when you get where you're going, turn it off, I think.
Well, there's di totally different levels there. The other thing I was thinking about is how about a or, an infrared light that you can look through the eyepieces. So I see you have one hanging from the wall back there.
Oh, those are my binoculars.
Yeah,
Yeah.
but they send out infrared and they change it over into visuals in your binocular. So that might be another way.
Absolutely. Yeah.
And if you've never been out to the Far East and seeing the incredible synchronous flashing there, um, I highly recommend it. You make your next vacation there because it is
Yeah,
really
it is.
fascinating. You know,
It's spectacular.
if you want to come by my house sometime and pick up a 1,000 feet of film, I can show them to you.
Awesome.
Stuck in my garage, naturally.
Yeah, so for those of you who don't know, so the synchrony that I showed, the video, I call it a loose synchrony because it's not like Christmas lights, right? And the fireflies in Southeast Asia, they are like Christmas lights. The synchrony is very, very close,
Yeah.
and it's stunning. They're
And they
also very,
flash very rapidly, as you are indicating, about every 800 milliseconds.
yeah, it's quite something. I see there's a question in the chat, so I'm going to answer that before it gets lost from Kelly. Um, the state parks have abhorrent lighting, no surprise, which I've been trying to get them to change. Is amber worse than white/blue?
No.
Well, so here's the thing. I think it's the color situation is always there's always going to be winners and losers. And there's many good reasons to not want a bluish white light because the blue part of a blue white light it bounces up and reflects around more in the atmosphere, so it causes more sky glow, and it also has more sort of human health impacts because our circadian rhythm and things like that are affected by blue light. And it also happens to be quite ugly. I think a lot of people don't like it. And so amber lights are pretty popular replacements for this. But to say that they're ecologically friendly, I think at this stage is a bit premature. Um, they because the reason I say that is, well, so one, they don't seem to be great for fireflies, and I care about fireflies, and so that's kind of, I'm biased here, but that seems pretty important to me. But even like not even discounting that, it's still very unnatural. If you think about a moonlit night, right? It's sort of, when you can see at night, the light that you see with the moon and the stars is kind of white. It's fairly whitish. Amber, like bright sources of amber light are pretty unnatural. And bright sources of blue light are unnatural too. So I think, on balance, maybe white is like is more natural and could be better for that reason. Although you could also argue using that logic that we should use green light because, if these are parks, we're talking about like the green light of the sun coming through the trees. But you can you can come up with a justification for almost anything. The point is, like, we don't have great evidence showing that changing color helps insects in any real measurable way. And what I do definitely notice is that people love to talk about color and they completely overlook things like shielding, dimming, and motion activators and timers. And those are way more effective. And so the first thing I always say is, so I say, don't worry about it and worry about the other stuff instead. Long answer, but, and then I think Eric's been waiting very patiently.
Yes, I had a question in regards to this too. First of all, what will be the impact not only on this, but just seasonal phenology as well, where it is based upon the length of day and night? And that's going to have a huge impact on a lot of diapausing insects as well. Have you thought about that yet, or is that a future direction, or I don't know?
I love to think about this. If I had more time, I would study this more directly. Luckily, other people study it for me, but it's fascinating, and I think it's the thing that we're all missing. Everybody likes to talk about the moth flying to the streetlight, and nobody's talking about the moth that doesn't go into diapause and then freezes to death
Yes.
because it's a
Yep.
lot harder to see. But so yeah, insects use light cues to decide when to do certain things, and they decide to enter diapause when the days get shorter. But if the light is making the days seem long, then they can make the wrong decision essentially. And there's a couple of studies that show this. It has to really like for a lot of species, for a lot of insect species, it seems like light is the thing they're paying attention to in the fall, and temperature is what they pay attention to in the spring. So the fall period seems to be the critical period. And, for an insect whose life cycle takes a month, right? If you're at like, if it's like October, November, you're deciding, am I going to diapause or am I going to give it another month? That's a dead end. If you don't diapause, you're likely not to make it to the next diapausing stage. So there's some papers about this. I bet it's pretty widespread, and it's fascinating. And there's also the effects of the light on the plant. It kind of goes,
Yes,
yeah.
that's what I was thinking too with phytochrome as well. With that, and thanks for bringing that up. Thanks for looking into that. And second of all, when you look at, is there any other way to monitor fireflies' activity and populations besides light? Is there any sampling method that's used? I've never heard of any, so
Yeah.
you can
So
see a change in populations. That's all.
yeah, fireflies, they don't fly to light traps, so you can't even do that. So this is, I think, why we have historically struggled to monitor fireflies. What you need are, it's really difficult because the best way to do it is, of course, to count the flashes. But if you have a species that flashes a lot, living with a species that doesn't, that has one flash every 5 seconds, how are you supposed to know? You have to know what species you're looking at in order to do this. And then there's just like an immense, like, I think there's an immense potential for community science to make something happen here because of all the interest. But at the same time, it's incredibly difficult to identify fireflies if you haven't been trained to do so. And so the thing that I think is most likely to work is this effort, which has been spearheaded by Orit Peleg at CU Boulder, Colorado University, where they use a set of GoPros, a 360-degree GoPro that can just record entire flash. What do you want to call that? A display, so it can do like a 30-minute display. And then they are developing machine learning methods of tracking the trajectories of individuals and stitching those into a flash pattern with its certain parameters that you would care about for identifying species, like flash duration and the space between flashes, and then using machine learning to sort of cluster them. And then you would do that, I mean, a lot of play, but you could imagine a standardized monitoring effort at one place over multiple years using the same cameras. You could start to tell some differences, probably, but it's tricky.
And I'll put in a final plug, sorry. Just to let you know, the West Virginia DNR is also monitoring different fireflies as well. So just to let you know. Thank you.
That's good to know.
Yeah.
Yeah, there's been it's, I think there's a lot of potential in it. But it's a tricky problem that continues to be tricky. So could light pollution drive long-term changes in mating systems for bioluminescent species? I think so. I think I think the given that light pollution is so patchy, probably the best strategy right now for a bioluminescent species is to go somewhere dark. If we run out of dark places, then I think that's maybe they need to start evolving some other things. But right now, I think movement is the key way in which most species cope with this. It's just the number of dark refuges are getting fewer and fewer. And then I see one more question here. Outside of color, is there a difference between incandescent bulbs and LEDs and how they impact insects, like attraction behavior, not just like attraction or behavior, not just overall light pollution? So the big difference would be that incandescent bulbs are very hot and LEDs do get hot, but not that hot. And so I think this does have an impact on their ability to fly next to the source. Anecdotally, I think, and for people here who do mothing, I'd be curious to know your thoughts on this, but I think a lot of the LED light lures that people use these days, you can get a lot of insects in the general area, but they don't really land or stay there. They sort of come and they hop away. Whereas I think some of the hotter, like the mercury vapor bulb, because it's hot, I think this really affects the insect enough that they tend to, I mean, and they also die more, right? They'll go get next to it and get burned. And so that's a big one. And I think, yeah, it would be an effective heat in some way, which that's the other big environmental resource, light and heat, right? So.
Have you ever done any mercury vapor lighting in the tropics? So I've
I
done
have
some. Yeah, you have not,
I have
or you have,
not in the
you have
tropics.
not. So I've done it in South America, and the attractions are unbelievable. At least it was when I went there some time ago and did that. Huge amounts. And of course, the mercury vapor light gets very hot. It didn't seem to bother them though. I mean, they came and they swirled around like for hours, and um, just an amazing amount of biomass.
Yeah.
Uh, they didn't really die, and at the end of the evening, we were ready to go. We had a big sheet that we shook after we took the light down. This cloud of moths just went up in the air. Uh, when you get a little slight rain accompany that, the attraction is even greater because
Mm,
they seem to come out with that little bit of rain.
maybe.
Um, of course, worst case scenario is what's happening now in the Amazon basin, like in Ecuador and Peru, where they have oil refineries or oil wells with 24/7 methane burnoff, and that flame just incinerates anything that comes near it. And the reports are you go walk around the base of these flames in the morning and you see zillions of carcasses of
Whoa.
big moths, beetles, giant beetles, all kinds of stuff that just gets scorched when they fly into the flame. So
What did you say is the name of that operation?
these are any oil well
Oil wells.
when they
Yeah.
pump the oil, there's a lot of methane that comes out that has to be burned off to avoid explosions.
Yeah.
So they have a 24/7.
You've cut out for me. I don't know.
Do you hear me now?
Yeah.
Yeah, it's a 24/7 flame that burns any oil well.
I guess I knew that. I never thought about insects, but of course, that's horrible.
Oh, it's staggering, let me tell you.
It's kind of amber.
And it's amber, environmentally friendly, famously.
No. Well,
Uh,
I'm glad that my students, I had a few who asked some questions of you, so that was good. Um, there's another question from a student here.
all right. Are there
Do
any
you see it?
surprising way insects adapt to or avoid artificial lighting depending on the spectrum or intensity of the light? Um, I think we do see that. Well, I don't know. Yeah, we in general, like UV light's really attractive to insects, which I think is interesting in itself because we don't really know why that is. and then they can't really see red or infrared at all. And that's kind of nice if you can use, yeah, for like looking at insects without them seeing you. That's quite good. But otherwise, I think most insects have pretty simple. I mean, I say most nocturnal insects, which are the insects that I care about, have pretty simple color vision, usually two photoreceptors. They're not doing anything crazy. The insects that have insane color vision are, the butterflies and the dragonflies. And in theory, they should be asleep at night, although dragonflies are very attracted to artificial lights, which is kind of a wild thing to see. I don't see, yes.
Well, we've asked you a lot of questions and comments and whatnot have come forward. And does anybody else have anything they'd like to ask? We have a large number of people have been hanging on here till the end. So if anybody has anything more they would like to ask, please do so now.
Yeah, I had something I just wanted to share. I've been looking at fireflies for maybe 15 years now. I don't have a PhD, but I've made some observations. One is that I think there's a difference between Photinus and what they'll tolerate and what they won't. Photinus pyralis, I've seen the males fly through sodium lights very nonchalantly, which I think is kind of interesting. And I've also tested Phochuris, and I'm not sure I knew what species it was at the time because it was said when there was this group, Firefly Watch, started by the Boston Museum of Science years
Yeah.
ago. They couldn't see blue light, so I put a few Phochuris, in a container, and I shone a fairly narrow spectrum blue light on them, and some of them scuttled, and some of them didn't care.
Mhm.
Okay, and same thing with UV. Some of them scuttled and some of them didn't care. So, and they were probably, one of three species. So that was sort of unusual because, I'm apt to test theories if it's easy to do it. So that was just something. Other thing is a firefly can see a red laser.
Yes.
They can.
Yes. So wow, I love that you're talking about this. So to go back to to go back to do I need to pull out the graph? I might as I'll just pull this one out. So so a firefly doesn't have a red photoreceptor. It has a green photoreceptor, which means it can't tell the difference between red, yellow, and green. It is red-green colorblind. To a firefly, they're all the same color. Because they have a green photoreceptor, the green photoreceptor is really good at seeing green, not so good at seeing red.
Yeah.
But if it sees red, it's going to think that it's green because that's the only color it can see. And
Right.
a red laser is really bright. So it's
Right.
going to look like a dim green light.
They also don't like green lasers. They're too bright for them.
It makes sense. That makes
Yeah,
sense.
it makes them go away. And I had a question is, can you post or send the articles that are referent to this? Because I'd like to look it over some more.
Yeah, I think if they're by me, you can visit my website. I'll put that in the chat, owenslab.org. And then if you navigate to there's a tab called publications, they're all linked on there. And almost everyone, if not everyone, is just open access. So you don't need any like permission to like read it. If you can't read one, just email me and I'll send you the PDF.
And there's an email link in there, I assume.
Oh yeah, on my website. Yeah.
Okay. All right.
Just to go back to so the thing about Photinus pyralis, I also love that you mentioned that because I think, yes, Photinus pyralis does seem to be really resilient to artificial light. There are reports of it flying around Central Park, down roads in major cities. And it's also the most successful firefly species. It's doing really, really well. It's expanding north, like it's everywhere. And I think this can lead people to think that fireflies are doing well because this one species is doing
Yeah.
well. This one species is also really, really dusk active compared to other Photinus. It comes out, you can still see the body flying around, whereas most fireflies come out like 30 minutes later at least. And so it's possible that because it likes to be active at twilight, we've created sort of an eternal twilight. All of these lights, it could be benefiting. And the other nocturnal species would then disappear, but we might not notice because people can't tell them apart. People don't know. But that's what I suspect is happening.
Mm. I call a pyralis the rat of the Photinus family because it's everywhere. Yeah,
Yeah.
it's the Photurus that I'm, more worried about at this point.
Yeah. Well, and a lot of Photurus are very nocturnal. So
Well, they sure are. Yeah, and the other thing I read, there was some article, and I can't remember. I bet you, uh, uh, Dr. Hansen knows that some of these Photinus have a pink filter over their eyes.
yes, yes. So that I think so the pink filter it is it's a blue light blocker.
Mhm.
So it basically they shifted from a green flash to a yellow flash. I talked about this a little, and they use a blue light blocker so that their vision becomes better and yet seeing yellow also. So it's sort of like putting on sunglasses so that they only see the colors of the others of their species. And yeah, we call them filter pigments.
I could go on, but I won't. Thank you.
That's fabulous. Thank you. Thank you for the comments. I love that.
Did you want some articles? I think you said you had a comment in the chat about posting some articles.
Yeah, I think we talked. They're on my website, so I put a link to my website.
Okay, good. Thank you. Is that the owenslab.org? Okay,
That's
gotcha.
the one. That's the
Great.
one.
I missed that. Okay. Anybody else have something they'd like to share or ask? And I want to thank everybody for
Oh,
coming out.
I've got a question, Fred.
Hmm.
Um, there's Plotinus that I'm struggling to think of the species name that comes out before Plotinus pyralis, and it's only found underneath the bushes and uh, shadows and places like that, and it only stays active for half an hour at most.
Interlands.
Scintillans? I was going to say
Pardon?
marginalis, but I don't know what species.
Marginal, that could be it. But at any rate, it comes out and then it goes back home sooner than Pyralis. Do you know if that's um, a light phenomenon in color phenomenon, I should say? Clearly, it's darker underneath there.
Yeah,
That
yeah.
we can.
I think we I haven't done enough on this, but similar with Photinus jamesi, formerly Photinus greenii, is a forest species, and it comes out earlier than a lot of other species, but I think it comes out earlier because the forest is darker before the field, if that makes sense. And
Mhm.
we did an experiment where we took what I would call, let me see if I can remember this. I think I would call what I would call a crepuscular, a dusk active species, a green eye, which is kind of in the middle, and then obscurellus, which is more nocturnal. We took these three and we put them in standard conditions,
Mhm.
and they all started flashing at about the same time. So I think it's an interaction between what their clock is telling them to do and then the habitat that they live in, where it's like if they start flashing when it gets so dark and they live in a habitat where it gets dark earlier, they'll start flashing earlier. But I don't know enough. I think it would be really interesting to kind of try to tease that apart.
Yeah.
Well, thanks everybody. I mean, if you have any more questions, I'm always happy to get an email from an interested entomologist.
What was it again? Owens at
owenslab.org. I'll put my, yeah, then it's
owenslab.org. Okay.
owenslab.org. Or if you just Google Avalon Owens Firefly, you'll get there eventually. Avalon's a pretty uncommon name. So
Yeah. Great.
So,
Well,
very
want to
nice.
thank you. That's very, very interesting, let's say.
Thank you.
Will you come back and speak again sometime?
Oh, I would love, well, first I got to come up with something new to talk about, but
Well, it won't be right away, but we'll get you back in the future if and by
I
then
would love that.
maybe you'll have something different to talk about or piggyback on something new that came along. So
That would be
it'd be
great.
nice.
Yeah.
I certainly want to thank all of our guests tonight. We had a wonderful attendance, 51 participants tonight, which is a great and you know, uh, folks like Eric from West Virginia and other people too. I think we had John Boken from West Virginia as well, and some other people. So great to see, and Dr. Hansen logged in, and um, what else can I say? I'm just very happy to have such a great attendance and lots of folks here tonight. Eric, I hope you join us again as well, and we'll have to communicate if you. Put your email in the chat for me. Maybe I'll shoot you an email sometime as well, if that's all right, and we can chat some more. So, um, Avalon, you're fabulous. Thank you for your talk, and um, look forward to seeing you sometime in the future in this small world that we live in, for sure.
And best wishes on being able to fund your stuff in these current difficult times.
Got to find a way to make it work, We'll figure it out.
Good,
Uh,
good for you.
thank you,
And I
everybody.
want
And
to thank you again for offering to speak tonight.
Mhm.
of course, thank
Great,
you for the
great
invitation.
presentation.
I really
Indeed.
appreciate it.
I,
Awesome.
can I give a critical comment in a good way?
Sure.
I've
Yes.
seen hundreds, if not thousands, of presentations like this, and you are so amazingly good. It was, it felt like you were an old friend to this group. Just
Aw.
something very personal, and it kept my attention, which at my age that's difficult to do. But I just think that the other presenters this year are going to have a hard act to follow. And
That's very
and
kind.
it kind of irks me to have to say this because the critters you study are the enemies of what I study. Your larvae eat the snails that my snail killing flies are depending on. But
Oh, an ecological interaction? Fireflies never have ecological interactions with anybody. That's fabulous. Wow. Well, I thank you. That's very, very kind.